Good afternoon, everybody. Thank you for joining us today in our webinar and our continuous medical education. And all the time, our topic is the current pandemic of COVID-19. And today, on behalf of Saudi Society for Clinical Chemistry, I would like to thank all the audience who's joining us right now. I would like also to thank our international speakers, well-known figures, Professor Giuseppe Lippi and Professor Mario Plepani, who's going to discuss this topic from a clinical side and also from the laboratory side. Due to some IT problem right now, we are trying our best to fix it. We have to shift our schedule, and we are going to start with Professor Mario Plepani as a first presentation. I would like to present our colleague, Professor Mario Plepani. He is a full professor of clinical biochemistry and clinical molecular biology. He is the past dean of Medical School of University of Padova, and he is the director of Department of Laboratory Medicine, University Hospital of Padova, and he is editor-in-chief of Journal of CCLM and Diagnosis, director of Center of Biomedical Research, Veneto region, and he is the director of Postgraduate School of Clinical Pathology and Clinical Biochemistry, University of Padova, from 2015 up to present. He received honorary professor of University of Buenos Aires from 2012 up to present, and he has published more than 1,000 original papers and exceeding 250 as a first author, and presented more than 500 abstract and poster communication. He received several distinction and awards in the entire career, including honorary diploma, Society of Biochemists of Serbia in 2017, and AACC standing contribution through service to a profession of a clinical chemistry award in 2019. Our topic, Professor Mario Plepani, will discuss the role of serologic testing in COVID-19 diagnosis. I would like to hand the mic for you, Professor Plepani. Thank you.
Good afternoon, everybody. My talk is about the role of serological testing in coronavirus 19 diagnostics. I'm from the Padova University Hospital, and I like to see you this picture dealing with the first anatomic theater in the world and the Aula Magna dedicated to Galileo Galilei in our old university. So my talk today is about the serological test, and I like to start why we have to use serological tests. We will move on the types of serological tests, both laboratory-based and point of care, the target of the assay, something about the importance of antibody kinetics, and what about the performances of the assays and the possible roadmap towards a harmonization, and finally, the testing strategies to be used in real practice, clinical practice, and final something about the humoral and cellular immunity. But first of all, I like to say you that we have to use the right test at the right time. And regarding laboratory medicine, molecular testing is for sure today the preferred method for the diagnosis of coronavirus disease. But serological tests play a relevant role, and I will deal with the role later on in my talk. And finally, many laboratory tests are essential to better understand the disease severity and for prognostication, and are clinical chemistry tests, hematological, coagulation, and enzyme that are very important. So, why we have to use a serological test? First of all, to improve our knowledge of the immune response to this new virus. Second, to better define and monitor the extent of virus spread in the population. Third, to screen particular subpopulation and to identify disease prevalence, for example, in healthcare workers for recovery and better understanding past exposure. To characterize efficacy of containment measures at the local and global level, the so-called lockdown, but also to screen convalescent sera for both therapeutic and prophylactic use, very important issue. To eventually combine molecular results for a more accurate diagnosis, and this is particularly in some difficult patients, to allow the diagnosis for later stages of infection, and in particular when the virus has been already eliminated. And finally, to identify individuals who have been infected but suffered only minor symptoms or did not seek medical attention. So many reasons to use serological tests. And which type of test can we use? We have two very common tests, point of care, that are generally lateral flow devices detecting IgG or IgG and IgM antibodies, or total antibody in different types of sample, whole blood, serum, plasma, and even saliva. Second types are laboratory-based tests, ELISA and/or chemiluminescent assays, which may detect IgG, IgM, and eventually IgA specific antibody separately or combined as a total antibody. And finally, we have the so-called neutralizing antibody that are very useful to determine the functional ability of antibodies to prevent infection of a virus in vitro. And there are two types of virus neutralization tests or pseudovirus neutralization tests, which are much more complicated. In this slide, you can see some advantages and disadvantages of both point of care and laboratory-based tests. Point of care, the advantage, less invasive because they commonly use capillary sample. They have a shorter analytical time, are easily implemented, but they have some disadvantage. The interpretation is very subjective. The threshold is fixed, and there is an analytical uncertainty, particularly around the gray zone. The laboratory-based present many advantages. Usually they should be qualitative or better quantitative results. They use venous sampling. They have high throughput, and the threshold, the cutoff, should be personalized. And this is very useful for the utilization in clinical practice. The disadvantage eventually is delay the results because the results are ready in 20 minutes or more if there are many samples to be analyzed. This is a picture from a recently published paper in which you can see the typical pattern of the so-called point-of-care testing, lateral flow, and you can see the type of pattern, and you can appreciate that sometimes it's not so easy to recognize the band. It's not so easy to say if the band is positive or still negative. What about the target of the diagnostic system, both for point of care and laboratory-based tests? As you can appreciate in this slide, there are today several laboratory-based tests, both in chemiluminescent or ELISA, and the targets are different. If we were looking to the Snibe maglumi we are using commonly in our practice, the target are both the S1, S2 spike protein and the nucleoprotein, and to detect both the IgM and the IgG specific antibody. For other diagnostic system, for instance, Abbott architect system, the target is the nucleocapsid phosphoprotein that is more or less the same as for the Roche Cobas electrochemiluminescence assay. The nucleocapsid measuring total antibodies, in this case, is a target. For the diazorin liaison, is a recombinant S1, S2 spike protein, measuring only IgG. For another assay that is a diagnostic system, DS, ELISA, as you can see, they are using the native antigen. There are a typical line of cells infected with the SARS coronavirus 2. Fortunately, some months ago, this paper provided us a protocol for serological assay, antigen production, and test setup. And I invite you to carefully read this paper that should be used by the manufacturer, first of all, but also by laboratory professionals to better understand the status of the art of serological tests. And this is a paper we have published with Professor Lippi on current laboratory diagnostics of coronavirus disease. And you can appreciate in this that we now know exactly the gene of the virus and the different target we can address the ELISA as well as the molecular testing. This is another more recently published paper in which you can appreciate the wild spike protein and particularly the receptor binding domain of the spike protein that is very important, and I will deal with this later on. You can appreciate that we know exactly the molecule, not only the molecule pattern, but even the different expression of this protein, and this is very useful to provide information to set up not only the antibodies, but even the total ELISA or chemiluminescent assay. So this is very important to be understood. Some months ago, we had some knowledge gaps in serological tests, because as you can appreciate in this slide, the kinetics of antibodies, the so-called time frame, was not so clear. And we have other knowledge gaps on the level and duration of immunity. This is still not completely known. And therefore, the likelihood of protection against the reinfection is still an open issue. And this is another reason to use a serological test to better understand the immunity and the immunity response to this new virus. What about the antibody kinetics? There are differences in the literature. We know the antibody targeting M protein and the RBD are the earliest to be detected, according to some references to some authors, in 100% of patients, at least 13 days after symptom onset. There are similar profiles described in the literature for anti-spike 2 antibody with a mean time lag of 2 days in comparison to the M protein and RBD, receptor binding domain. And finally, antibody targeting the S1 subunit seems to be the last to be detected. So not an early kinetics, a late kinetics using this type of target. This is one of the first papers published by K. Wong To in latent infection disease. You can appreciate in this slide the behavior of IgG and IgM having target the receptor binding domain. And according to this picture, IgM do not appear early than IgG in some patients. That is an unexpected issue. This is a more recently published picture in which you can appreciate the time frame of the antibody in comparison to the detection of RNA of the coronavirus 19. So there is no doubt the molecular test is an early message and detection time in comparison to the antibody. Second message, total antibodies, in this case provided by ELISA, but even by other chemiluminescent agents, appear early, and thereafter the IgM, and later on the IgG antibody does appear in this in the patients, in affected patient. So, summarizing, it seems that total antibodies appear a little bit before IgM and before a little bit early than IgG specific antibody. And you can appreciate the time frame in which you can see that only few patients present the antibody between 5 and 10 days. Much more patients present antibody after 10 days and between 10 to 15 days, and maintain the level of antibody later on. So the step to be performed when evaluating serological assay for COVID-19 disease are, first of all, to validate the analytical performances in precision, repeatability, dilution, and eventually recovery studies. Second, to understand the seroconversion time, the kinetics and the rate. Third, to better understand the diagnostic specificity and sensitivity of the assay, and much more interesting, the predictive positive and negative predictive value, and finally, the use of the test in epidemiological studies. And this is what we have done, starting from the evaluation of the first IVD chemiluminescent assay for SARS coronavirus 2, that is the Maglumi by Snibe. As you can see in this paper published in Clinical Chemistry Laboratory Medicine, you can appreciate the very, very nice repeatability and intermediate precision with values lower than the maximum is 6% of imprecision, quite good. It was evaluated according to the traditional recommendation by the Clinical Laboratory Standardization Institute. Here you can appreciate the linearity, the study of linearity, which demonstrates a very, very good linearity of both IgM and IgG as measured by Maglumi. And the recovery results in percentage are quite good. So from our point of view, this means that the Maglumi-Snibe should be defined a quantitative, not only qualitative method for measuring specific antibody for coronavirus 19 disease. And thereafter, we have evaluated the kinetics. And you can appreciate in green the kinetics of IgG, and in blue the kinetics of IgM. As you can appreciate, in some patients, they are starting 6 to 8 days after symptoms. And in this case, the symptom was fever, the first symptom. And IgG are maintained for a long time, while there is a decrease of IgM after 18, 19 days. You can better appreciate the kinetics in this table, a descriptive statistics of the kinetics, in which you can see that between 12 to 13 days, 100% of subjects or patients developed IgG antibodies, detectable IgG antibodies, while the IgM were present in more or less 80% of the patients. In this paper, another paper, we have evaluated the IgA antibody with a test measuring the spike glycoprotein of the coronavirus. You can appreciate in this spaghetti plot the behavior of the IgA on the left and the IgM on the right side to better understand the kinetics of the different specific antibodies. And you can better see in this slide the kinetics of IgA, which seem to be to give us an earlier indicator than IgM, and they are more or less stable. Even there is a not significant but mild decrease after 40 days, starting from the onset of the disease. So, after these studies, we have started to better understand the roadmap towards a rational use of serological testing. And first of all, we have to better understand the presence of the antibody in the population. That means the prevalence of the disease. The second point is understand the performance of serological tests, both the ability to detect the presence, the so-called sensitivity, as well as to confirm the absence of the disease when they are truly absent, when the antibody are truly absent. That means specificity. Third point, we have started to collect data whether and how antibodies confer immunity, the degree and persistence of the immunity. And finally, we have to balance the net harm of mistakenly release a susceptible, potentially infectious person, the so-called false positive cost, and the net cost of failing to certify a truly immune person to rejoin the workforce, the so-called false negative cost. So, starting from the beginning, this is the first time I'm presenting this data. The paper has been submitted but not yet published. These are the results of a sero survey in healthcare workers of the Veneto region, particularly in the hospital of Padova and Verona. As you can see, the percentage of positive tests is around 4%. There is a little but not significant difference between the different age classes lower than 30 years, between 30 and 39, and so on, older than 60 years. And you can appreciate that the percentage of positive tests is ranging from 3.5 to 6%. So the mean value is 4%. The table 2 shows the total number and the percentage of positive tests with the 90%, 95% confidence interval subdivided by the different healthcare figures. And you can appreciate that the percentage of positive tests range from 3.6% physicians to 4.7% in nurses to final 6% in healthcare assistants. And this difference is statistically different, reach statistical significance. And this is another very interesting result, the prevalence by disease severity. So antibodies were found to be present in all healthcare workers with a severe disease, which means that they were all hospitalized. The antibodies were present in 83% of healthcare workers with mild disease, and finally only in 58% of totally asymptomatic subjects with virus demonstrated disease. So this is very interesting because asymptomatic patient seems to mount the specific immunoglobulin only in half of the healthcare workers with an infectious disease. In this paper, we have evaluated the diagnostic performances and threshold of 5 different assays for antibodies detection. 3 are chemiluminescent assays, including the Maglumi Snibe assay, which are using in our clinical practice. There are other two chemiluminescent assay and two very commonly used ELISA assay. In this slide, you can appreciate the behavior of the IgG starting from the left side on maglumi, in which you can see the differentiation between mild disease, moderate disease, and severe COVID-19 disease, with an increase of the amount of detectable antibodies, IgG, and in comparison to negative healthcare workers, autoimmune disease, and sera from donors before with the sample before 2019, that means before the appearance of the new virus. You can appreciate the distinction between the patients and the negative subjects without the disease, without the virus. Vice versa, this is the behavior of the IgM antibody, in which you can appreciate that the difference between mild, moderate, and severe patient in comparison even to negative is not so good as previously reported for the IgG. Thereafter, we have started to better understand what should be done to improve the agreement between different assays by changing the threshold. So we identified the best threshold cutoff, providing 95% of specificity. And as you can see, the agreement between the maglumi Snibe and all other assays is very good after changing and optimizing the cutoff. And this is very interesting data. You probably know the Fagan nomogram, in which starting from a pre-test probability, in this case with a prevalence, a low prevalence of the disease, 4%, exactly the prevalence we have detected in healthcare workers in Veneto region, both Padova and Verona hospital. You can see that the negative predictive value using the maglumi is very good. If the result is negative, we can say that at 99% there is no infection disease detectable in the subject, even with the low prevalence of disease. Using the same probability, 4%, the predictive positive values is not so high, is more or less around the 50%. But if we are using a higher disease prevalence, in this case 23%, which is a prevalence described in the literature in other areas of Italy and probably in New York area, you can see that the predictive positive value with the maglumi reach a very, very high level, more or less 90%. So while the negative predictive value is always very good and there is no influence, detectable influence of the prevalence of the disease, with the low prevalence of the disease, the predictive positive value is not so good, but not only for the maglumi, for any other serological tests. We are moving to the point of care testing now. The major issue is the reader training. because it was already described in the literature that is necessary to have a good reader training as a key to have reliable performances. The agreement between the point of care, in this case is the lateral flow tests, with those of IgG and the IgM epitope detected with an ELISA ranged from 75 to 85% according to the different types of device used. And the band intensity score show direct correlation with the ELISA signal and the cutoff. So this is very interesting paper, demonstrated that particularly with some, not all, point-of-care testing, we can achieve good results. But if you look to this picture, you can appreciate that not all point-of-care testing are working well, and the agreement with the reference method, in this case the chemiluminescent or ELISA assay, is not perfect, is not good for many point-of-care tests. So what we have to do to provide valuable testing strategies? The aim is to maximize specificity and therefore the positive predictive value, PPV, particularly as the overall prevalence is likely low, as in general population. So the strategies recommended by the Center of Disease Control in the US are, first of all, choosing a test with a very high specificity, 95% or greater. And this is why I prefer to use chemiluminescent or ELISA test, in any case laboratory-based test, because you can modify the cutoff, the threshold, to obtain this type of very high specificity, 95%. The second is to focus testing with high pretest probability. For instance, in subject or patient with the history of COVID-19 illness. In this case, we have to give access to the test only to subjects with high pretest probability, and this seems not to be good. Finally, to employ an orthogonal testing algorithm, that means to test with the second assay any positive person with previously tested and with a positive result. And this is largely recommended in the US. I'm not so sure this is the best strategy. In my opinion, the best one is to choose a test with a very high specificity, that means 95% or greater, if possible, to have a specificity near to 99%. Recently, this paper published in Nature discussed the value of serological test with a very provocative title, 10 reasons why immunity passports are a bad idea. In my mind, I can deal with the 10 points. The first one is the coronavirus 19 immunity is a mystery. The second, the serological tests are unreliable. The third, the volume of testing is unfeasible. Too few survivors to boost the economy. Monitoring erodes privacy. Marginalized groups will face more scrutiny. The seventh is unfair access, societal stratification, new form of discrimination, threats to public health. I may say that at least 7 of these points are absolutely not understandable, or I cannot agree with most of these points. And therefore, if coronavirus 19 immunity is a mystery, and this is true, or it was some months ago, this is a valuable reason to use serological tests to improve our knowledge on the immune response to this new virus, the coronavirus CoV-2. The second point, they say serological tests are unreliable, but an increasing amount of data are available to demonstrate the accuracy and reliability of some commercially available tests, particularly some chemiluminescent assay. So this statement is absolutely wrong. Now we have data and evidence to demonstrate that many commercially available tests are quite good, are reliable, and accurate. What about the humoral and cellular immunity? We know that SARS coronavirus 2 specific antibodies are detected in convalescent subjects. We know that most convalescent individuals have detectable neutralizing antibodies, and these antibodies should be used to treat new detected patients. We know that cellular immune response, both B and T cells, to SARS coronavirus 2 are found in COVID-19 convalescent subjects. And finally, we know that neutralization antibody titers correlate with the number of virus-specific T cells. So this is a very, very interesting point. I will show you this very recently published paper by Grifoni et al. In Cell, in which you can detect the difference between COVID-19 subjects and patients, what happens to CD4, CD8, and other subpopulation, sub-lymphocyte population, the difference in regard to unexposed patient to the new virus. And in this slide, you can appreciate the difference in several types of T cells and B cells in response to the coronavirus infection. So this is a very interesting research topic, and it is very important because this opens a way to better understand how we can provide new vaccine platform. You can see in this slide that starting from the spike protein and the other protein already recognized, and particularly the receptor binding domain, there are several strategies to develop new vaccine candidate, and this is not only in the preclinical testing, but as you know, we are very near, according to the information we have, to have in 6 to 18 months very actionable vaccination projects to help us to fight the disease. So this is the virus. Some months ago was totally unknown, but now the pandemic has given a clear example of the critical role that the lab plays in healthcare system. because the laboratory is a cornerstone for the diagnosis and monitoring of the disease. The value of the lab cannot be held at the cost per test level, but should be measured in the downstream impact, and in this case, accurate, reliable, and timely results that are used not only by our clinical colleagues, but also by policymakers as they plan strategies to mitigate this pandemic. So I like to finish saying that we have clinical labs play a key role in fighting the new SARS coronavirus 2, and I'm sure that we will totally defeat the virus in the next few months. Thank you for your attention.
Thank you so much, Professor Plepani, for your excellent presentation regarding the current updates regarding the technical performance of different assays at the lab for COVID-19 testing and point-of-care testing. And just I would like to announce to the audience that Professor Giuseppe Lippi has some technical problem in his PC. So so far we couldn't get him in with us during the webinar today. And we are ready now to with Dr. Plepani to take some questions. We can read some questions from the chat with the attendees. So I will select the first question that from Mr. Mustafa Sawas, that he is writing that, is it possible to get a positive PCR results for COVID-19, then after 20 days the result of IgG is negative?
After 20 days?
Yes, and then we get the IgG negative.
I have to say that in some particular patients, I mean patients treated with antiviral therapies, the kinetics of IgG demonstrate a decrease after 1 or 2 weeks. This is only in particular selected patients. So the answer is yes. In some patients with positive molecular testing, after 20 disease, the kinetics of IgG should present a decrease. But this is not a common behavior. Mm-hmm. Most of patients and infected subjects, even asymptomatic, have a permanent presence of IgG over at least 60 days. I cannot say anymore because we started to measure the antibodies more or less 60 to 70 days ago. But yes, there are some particular patients in which we cannot understand why the kinetics, the decrease of IgG is earlier than in others. And probably this is an effect of the therapy, and we would like to study this particular type of subject and patient because nobody knows what happens, and if later on the IgG increase again.
Thank you, Professor Plepani. Our second question from Dr. Muhammad Iqbal, that his question is the immunity produced in COVID-19 is mostly proved to be infection immunity, not prevention antibody. Hence, there is a high possibility of reinfection even after the antibody production.
Oh, this is a totally unknown issue because only there are some trial up to now using monoclonal antibody to prevent the disease, and in some animal models it seems that they are working, but we cannot know in the humans. So this is I cannot provide an answer. I cannot provide how long the immunity in this case should be maintained over time, because it's too early. We know that, but most of the data are in favor of maintenance of the immunity, at least for SARS and MERS, that means 24, 30 months.
So thank you, Professor Filipani.
But the problem is that for the neutralization antibodies, there are some difficulties in both the technical and in understanding if they are really blocking the virus. It's not easy to have the data up to now. So it's still in progress.
Okay. Thank you, Professor Filipani. And we have a question. Disappeared. Sorry. We have a question is from and just is just related to the management from Mr. Khalid Ouda. I think he's asking general question: what is the relationship between COVID-19 and hydroxychloroquine? I mean, the current what has been discussed as a treatment. So this is a general, I think, related to the clinical and management side. If you'd like to give feedback, Professor Filipani.
Is a very controversial issue. You know that the paper originally published in Lancet was withdrawn because there were some problems in the statistical treatment and the comparison. So if I have to say you the opinion of my immunologist and the colleagues of infectious disease in Padova, it seems that this therapy is working. I don't know if this is working because they are using the right dose in the right subjects or patients, but they obtained very good results. Looking at the literature, this is a controversial issue, as I said before. So no definitive answer, but I may say that my colleagues in Padova University Hospital would like to continue to use this type of treatment in comparison to other antiviral therapies because they obtained very good results.
Thank you, Professor Filipani.
It's not my field, but I'm working with the colleagues, and so I can spread the information I collected.
We have a question from Mr. Shawqi Farouk, that which is a general question related to which is better for plasma donation, presence of IgG or IgA in the donor.
No. For plasma donation, we have to have very high level of IgG with neutralizing function demonstrated. So there are two essential issues. First, high level of the IgG, and second, the evidence that they are neutralizing the virus.
Okay, thank you, Professor Filipani. We have a question from Mr. Nawaf Al-Utaibi, that he is asking that why we are using real-time PCR, not PCR.
No, we are using this is another real-time PCR because it is much more easy, and we have the instrumentation as a method originally developed and recommended by the World Organization, was based on RT-PCR. There are digital PCR, there are other types, but the much common type of molecular testing is real-time PCR. Reverse real-time PCR is widely used, but
thank you, Professor Filipani. And just I'm trying to overview certain questions. We have a question from Dr. Samia Subki regarding the the serological conversion, is there is any difference between different population, for example, Asian versus European?
No, the unique difference up to now is, of course, the immunodepressant subjects cannot mount immunoglobulin, or to a lesser degree that immunocompetent subject. But there are no evidence of gender or race differences. So no data about this, but I don't think that is the immune response is very similar.
Thank you, Professor Plepani. And well, we have a question from Astrid San Juan. That what is the best time to test for antibody in asymptomatic patient? An asymptomatic patient.
According to the kinetics we have evaluated, only after 12 days, even for asymptomatic subjects, this is a time for better understand the behavior of the detectable antibody. In our experience, after 12 days.
Thank you, Professor Plepani. Over that some of them answered during the presentation. I think there is no need to go back over them again. I just have a question, Professor Plepani, regarding, you mentioned the point of care testing.
Yeah.
So what is the current from your experience right now? What is the current utilization based on algorithm of testing for point of care testing?
You know that the main issue, the advantage of point of care is the use of capillary sample. So think about the children or other types of population. In our hospital, for instance, we are using for diabetics that are already collecting the capillary blood for glucose, for immediate measurement of glucose, even a drop for the antibody testing. So in my mind, this should be used when the subject is children or other types of patients who prefer to have the capillary blood sampling instead of the venous blood. For what is strange is that if you are looking in the literature, the description of the performances of the point of care is mostly provided in venous blood samples collected with the DTA. And this is very strange because in my mind, the advantage is the rapid, the sampling, capillary sampling, and rapid results after 15 minutes, delivery of the result in front of the subject. For all other things, I prefer laboratory-based tests, of course. And the reason I said is the evaluation of the performances, but much more, we can identify the cutoff which provides the 95 or 99% of specificity. This is very important. And this is impossible with the point of care because the cutoff is fixed, is manufacturer based, and you cannot ask the manufacturer to change, but this is impossible to be realized, of course.
Thank you so much, Professor Mario Plepani, for your excellent presentation and the current highlights regarding the testing algorithms for COVID-19 right now worldwide. And we would like to apologize from you and all the attendees and audience today with us in our webinar regarding the technical issue we faced with Professor Lippi, and we hope to see you in the upcoming and next webinars with the Saudi Society for Clinical Chemistry. I would like also to highlight that regarding all the audience and attendees, they will receive email from us just regarding a survey and some data they should fill it up for the Saudi Commission in order for continuous medical education hours to be credited in their account with the Saudi Commission for Health Specialties. Thank you so much for your time. Thank you so much for your highly informative presentation, and we pray all together to save the humanity and to eradicate the COVID-19 very soon, together worldwide. Thank you so much, Professor Plipani. Thank you.
Let me say I'm delighted to have this opportunity to deliver this talk. I like the Saudi Society Clinical Chemistry. I have to thank you, the Snibe, for the kindly supporting this webinar. And my message is we have to continue spread and share the information about the laboratory testing in this area as well as in other areas, and to have a common view of the future work in laboratory medicine, because it's very important to share our information, knowledge, and improve the quality of laboratory testing worldwide. So thank you so much, Ali. It was a real pleasure to be with you today. I'm sorry for the technical problem with my friend and colleague, Professor Lippi, but we will have other opportunities to join you
very soon.
and to share with you our knowledge. Have a nice day.
Inshallah. Thank you, you too, and everybody else.
Ciao.
Thank you. Ciao.
Ciao.